The Zero Input Agriculture podcast is a solo passion project built with my ample time and meagre resources. If you would like to support the show, then sign up for a paid subscription to my Zero Input Agriculture blog at Substack. As a reward, you can read all my past posts and listen to the audiobook of Taming the Apocalypse. If you prefer reading about the past and future of biological technology, you can read the ebook of Taming the Apocalypse through Amazon. And finally, please rate and review the show on whatever podcast app you use, since it helps other people find me. Or recommend the show directly to anyone who could use some inspiration to become a plant breeder themselves. Welcome to the Zero Input Agriculture podcast. Today we have Eugene McCarthy back for a third time because I am so enchanted, I think is the right word, by his theory and its potential applications and implications. So that's the topic we want to focus on today. If it is true that hybridization is a major driver of speciation, particularly that wide hybridization is much more possible than we previously appreciated, what does that mean for the future of biological technology, of how we study our past, and what we could achieve in the future? So welcome back, Eugene.
Well, hello. I'm glad to be back. I enjoyed our previous chat, so I suppose this one might be enjoyable as well.
I definitely hope so. So let's start off by just recapitulating the the implications of hybrid based theory for the study of the past in terms of like phylogenetic reconstructions. Like, where do you see that whole field going in the future if it embraces the idea that hybridization was important on all sorts of different scales in the past?
Well, if you're talking about taxonomy and phylogeny and essentially the process of ordering life into a tree-like structure of classification and a tree-like process of descent, then um, really all of that would be pretty much invalidated, or just would it would just not be useful except to say perhaps a filing system. It would have it would not be needed, I guess is basically what I would say, because if the actual process, if as I've argued elsewhere, it's that it's reasonable to think of the typical new form of life as coming into being by means of a stabilization process that is typically triggered by hybridization, then you don't have a tree-like process that you're describing. You have a lot of different types of organisms coming together, mating, producing offspring that are of mixed heritage, and then these populations may or may not get established over time if there's sufficient stability, if the traits that they have are sufficiently favorable, then they may get established, or they may just keep on getting produced as hybrids over time by their original parents, which happens a lot. There are say hybrid zones here in the United States where like there's the Baltimore Oriole and the Bullock's Oriole. They come together in the central United States. They have this long hybrid zone that stretches from Canada down to, I think, Texas, and it's about 200 miles wide. And all of those birds in there are hybrids. And virtually all of them are later generation hybrids, but they're not getting established as a new type. They're just being produced on an ongoing basis. But then you have other things where they brought to New Zealand and started breeding with the Pacific black duck there back in the 1800s. And now apparently the black duck and it's getting just kind of wiped out there. And then all the ducks there are like mixed heritage. And eventually you may have a new type stabilize out of something like that. So I think in that situation, it helps as this island where it's not continuously getting a new input of mallard. It's just kind of isolated from other areas, and it would help that process. And letting them get established. But there are all sorts of different methods by which these stabilization processes can occur. Like, could be that you produce a polyploid that's reproductively stable, and that can happen just in a generation or two. And it can also happen repeatedly as you do the same hybrid cross, you keep getting these polyploids out of it. There are other situations where you have a couple of parents that cross to produce an asexual organism, or these asexual lizards. I'm thinking of, I think it's Neomexicanus, southwestern United States, where they are immediately stable because they are immediately able to, they're all female and they just reproduce themselves clonally.
Mm.
So I think that people think a lot about with hybridization that you're going to get something sterile, which even that is not always the case. But even when you do get something that's sterile, in many cases, in other words, what I mean by sterile is unable to produce gametes, sperm or eggs. Even when you get that, you can get organisms that can reproduce vegetatively or by other means than sex. And
Mm.
so, I mean, I think it's 98% of all invertebrates can reproduce without sex. All the different 98% of all the different kinds of invertebrates are the vast majority of animals
Hmm.
or invertebrates.
Do you think we need to refine the concept of extinction?
Redefine it?
Yeah, well, an example is Neanderthals are extinct, but there's more Neanderthal DNA alive in modern humans than there was at the time that there were only Neanderthals in Europe, for example. So are Neanderthals actually extinct on a genetic level? I mean, there's probably some of their genes that have been selected against and lost from the population, but
Well,
in some ways they're living inside us now.
well, I think a lot of Shane of the way people think about things is kind of typological, I guess you'd say, is that they think, well, I find some bones here in Europe, and that's one kind of organism, and I find some bones over here in Russia, and that's some other kind of organism. And I find some other bones in Indonesia, and that's some other kind of organism. But if you actually went back to the time when all these various bones were still alive, there might have been a more continuous population, and this might be just a lot of the types have arisen just out of the paucity of specimens and the isolation of the locales and the distinct researchers that are working on the projects, and they want something that's different because they're not going to get nearly as much attention publishing a paper about something that has the same name as something that's already been found as they if they like come up with some new
Yeah, yeah.
something new, there's some new name, new so-called species or whatever. Um, and when they do that, they make a splash. And so there are all these factors that are beyond just the genetic that contribute, I think, to the idea of these various types that are proposed. It's not just the anatomy, not just the genetics, but it's also these other factors I was just talking about that lead people to think about these things as distinct. Where if you went back and you had a very large sample size, it might be that you would see there were lots of intermediates between these existing at the time that they were alive. I mean, even now, if you want, say you imagine like a million years from now, someone went to dig in the Congo area, they would find these small people, and they say, "This is a distinct species. We found
Hmm.
something really cool here. This is really neat." They're these small little hominids, but I don't think they're considered by us to be anything other than human beings. At this present time, they're just a variety of human beings, and so it may be that a lot of what this, these descriptions, like you're saying, or Neanderthal say, it's could just be a lot of it reflects that. And then also, if there are different types, you can have obviously all kinds of hybridization going on between virtually any type, not just between humans, but between humans and non-humans and other types of all kinds of animals all over the earth. All sorts of plants are hybridizing with other kinds of plants, and so you have all sorts of intermediates. And so, do the intermediates count if you cross A and B? Do the intermediates count as an example of A or an example of B, or are they something separate, or are they something in between, or what? It all depends on the way you want to think about it, I guess, is what I would say.
Do you know, I have a prediction about what academic phylogeneticists will do in the future. So just using tree-based models, they're pretty much getting to the end of the project. Like they've analyzed pretty much every major group, put it on a tree. They shift the branches around every now and again, but it's kind of becoming an old story already. If they absorb the hybrid-based model, they've got an opportunity to publish so many more papers, trying out so many more mathematical approaches to analyzing this mountain of genomic data to try and figure out where those hybridization points were. And I can actually see them having an incentive probably in the next 5 years to say, oh yeah, it's not a tree, it's something completely different. Give us more grant money and we'll figure out what the branching structure looks like.
Yeah, well, the thing is that it gives you a better idea of exactly what the organism is if you can identify its parents. But it also, if you're talking about going back far in time, it muddies the waters for that, I think,
Hmm.
because it, it becomes, well, so let's put it this way. You can take any set of objects with a variety of characteristics, and then according to how many characteristics they share, arrange them into a tree. If they share more characteristics, they're closer in the tree. If they share fewer, they're further apart in the tree. Say, I think the example I've used with you before is like the various things in my basement. You can have the washing machine and the dryer. They're similar objects. They would go close together in the tree, but that would not be as close to, say, my power saw. It would be like off on a more distant branch, and
Hmm.
then my furnace would be like in a still more distant branch, and then the sump pump might be off as an outlier. But that doesn't mean that all those are descended from some common ancestor just because I can arrange them into some sort of tree. That doesn't. That's just all an assumption that is in the mind of the person that's arranging the tree, it, and if hybridization, as I'm suggesting, is typical in the production of new types of organisms, then the tree-like structure just becomes a figment.
Mm.
It's not something real or something that can be determined. And also, if you do start determining, you use different data sets, you start getting different results from different data sets. If that's the case, that's the real picture, then but different phylogenies will produce
This brings
be
up a
produced
deeper
from
question.
different data sets.
Like it brings up a deeper question. Like, what are phylogenetic trees for? Are they just a satisfying story that we like to tell ourselves about the past? Are they like a creation myth for the Asteraceae?
Well, I think originally, like back before Darwin, I say back at the time of Linnaeus, it was a system of classification that went along with this Aristotelian outlook where you had categories and then you had distinguishing characteristics that you would combine, and then you could arrange things into this tree-like structure of classification. And then Darwin came along and suggested, aha, this means something about history.
Mm.
This classification system we have, it means something. It's just reflecting a history that we haven't realized before. But in my opinion, that probably is just not the case. It's probably was just a classification system, and Darwin was mistaken on that. I think that he was. I mean, it seemed like a profound insight, but that happens a lot of profound insights. It turns out to be a mistake.
The only way I could see it being useful is if we had a time machine and you're like, I want to go back to where, this family appeared and you have to set the coordinates to like the right time and place, but we don't have that technology. So it's kind of it's a moot point as to exactly how a particular organism came to be.
Yeah, well, when you say it, when you say it is useful, what are you saying is it?
Well, I guess at the narrowest you'd say a particular like plant and an individual plant, and you could trace it back to its original ancestor, like one generation back or 10 generations back, you could keep those records. But the further you go back, the fuzzier the data becomes.
Yeah, okay. It's like my favorite Tacitus quote. It's like all antiquity is obscure. I think that's true. I think that's true. It's like people like to imagine that we know just what was happening a million years ago, say in Asia or whatever. But
Mm.
I don't think we really do know a lot about what happened a million years ago in Asia. We scarcely know what was happening, like 3,000 years ago in the Middle East. I mean,
It reminds
we,
me of a lovely talk I saw by a geopolitical analyst, Samo Boria, who pointed out that we have this common modern notion that as time goes on, we learn more and more about history. And he said, no, it's actually the opposite. Like history is constantly decaying. Like live accounts disappear when people age and die. Documents get like destroyed and shredded and damaged. And even artifacts from like long ago are constantly being lost and damaged and misplaced and misclassified. So, yeah, as time goes on, history becomes more obscure.
yeah, and obviously the further back it goes, the more obscure it's going to be.
Mm,
So I think Tacitus was very right. But at the same time, this I think the hybridization kind of outlook that can be very informative. I think that in the case of humans, for me, it's been very informative. It's like I before looking at it that way, I would never have supposed that we might be hybrids derived from the type of cross I've been talking about, which the evidence all points to something like bonobo cross some kind of pig
hmm
with back crossing to bonobo, probably a
Oh,
couple of generations of back crossing. So to, that's very interesting to me. And I think to a lot of other people I've spoken to, that's very interesting information, but it still doesn't, trace back and tell you where bonobos came from, where pigs came from.
mm.
Although you might make some progress thinking in that way, of thinking about it in that way. But
To me, the more interesting thing is what can we do with these ideas now leading into the future? Because they suggest that there are possibilities in biology that we are not even trying because we assume, from basic principles that they're impossible. But, it's kind of a circular argument. It's the negative version of the chicken and the egg. No one's willing to even start testing out these ideas with the current worldview that's dominant.
Mhm. Well, I do think that it's, I mean, to a certain extent, it is used. I mean, you have things like, say, hybrid corn. I mean, there is this idea that you can cross things, but I do think that there's an idea that when you get more than a certain distance between the parents in similarity, that you're not going to get anything. And
Mm.
so in those cases, they don't try. But I think that that's wrong that to not try. For example, this guy at the University of Chicago, H. Newman, back in the early 1900s, was just taking all sorts of different fish and, some distantly related, some like separate super orders, and just squeezing out the eggs and the milk and mixing them and seeing how many of the eggs were fertilized. He got lots of results doing that. So it could be that people took the same mindset and were doing it, say, with something where it was fairly easy to do cross fertilization. Like it's pretty easy with a lot of plants, say, to just take some pollen and paint it on another flower, and it's not hard to get cross fertilizations going. And you can perhaps find out a lot of new crop plants that way. But if nobody's doing it, you're not going to find it out, of course. And then if you have the attitude that you can't get anything, then nobody's going to try it. So it's just like you're
Yeah.
saying, it's just kind of a circular thing that self-defeating.
Mm.
But
Focusing on the vertebrates, could you imagine? Let me paint a picture of a collaborative research project. So we've got different labs working with different model organisms scattered all over the place, and you could and they're dealing with the technological manipulation of the reproductive cycles of those organisms. So they're doing artificial insemination, in vitro fertilization, implantation, all that kind of stuff. And I could imagine a network of like 10 of these different labs, each working on a different organism, gathering sperm, freezing it, putting it into portions, and then sending it out to all of the other organizations to try doing in vitro fertilization. Because often the biggest stumbling block is just in that early formation of a viable embryo. And if you do that with a mass of like induced eggs, in vitro, you can then screen them for the ones that have the right lucky combination of genes to be viable
mhm.
for implantation and then further development. So that would potentially reduce the number of like dead ends that you'd go into. Like if you just did artificial insemination, I think you get a much lower success rate.
Yeah, well, it's true that you could use that sort of approach. I'm not sure. I mean, I'm not myself a breeder, and I have not myself, worked. I read a lot about what breeders do. I've read thousands of papers and books on what breeders do, but I don't do it myself, so I don't have a good sense of what's efficient in that regard. But it's true that you could try everything via some process like artificial insemination, but there might be other things that work too. Like one thing that's fairly easy is just to put animals together that are likely to mate.
Hmm.
Um, and then they carry out all the work for you, and they do repeated inseminations and sometimes get pregnant. And sometimes, the results are viable. But the thing is, like, if anything that's done in a laboratory setting is a lot more expensive than, say, on a farm or
Hmm.
on, the sort of things, the sort of setups that breeders have. And so it might be, I'm not the one to judge the economics or the efficiency of it, but I have the feeling that, say, if you wanted to produce a particular type of hybrid from, say, two types of animals, the thing is to get those two animals together in a situation where they don't have much of a choice to mate with anyone else.
Hmm.
And then they will mate with each other, at least in a lot of cases. So there's some animals that have much more of a propensity to mate with other kinds of animals than others. Pigs, say, example, are one. Dogs are another. Ducks are another.
Hmm.
Also chickens. So those four animals will mate with a tremendous number of different kinds of animals. Like I have like records of probably over 100 different kinds of birds that chickens have mated with that are not chickens.
Hmm.
And so like dogs will mount virtually anything. Birds, they'll try to mate with humans. They'll try to mate with almost anything. And then there are other kinds of animals that have much more persnickety, and they don't really want to, they're much less, they're not indiscriminate in that way, though they're more particular. But even those, if you expose them to the proper early conditions so that they're imprinted on some other kind of animal, they will do it in that case too. So it might be that a pretty efficient method would be, it depends on the particular organism you're working with, but organisms that are very susceptible to imprinting.
Do you have any thoughts on what the limits of wide hybridization might be? We were kind of talking about this before. Do you want to sketch out that graph you were talking about?
Sketch that graph out?
Y-
Well, I was mostly bringing that graph up as a negative, and since we were talking about that before we started recording this session, I'll
mm,
describe it again. It's
yeah.
say you have a two-dimensional graph that you're constructing, and the x-axis looks like as you went from left to right, it would be along that axis would be interspecific crosses, or I mean, even you can start with inter-subspecific crosses, then interspecific, intergeneric. Just look at their taxonomic classifications and interfamilial, keep on going to the right, and interordinal, interclass, interphylum. Uh, to actually give empirical weight to the statement that more distant crosses are less likely to work, you would have to show that if the y-axis on that graph was success rate of inseminations or matings or pollinations, whatever you want to say,
Mm,
then that plot should be going down as you go to the right. If you, for an equal number of trials,
yeah.
each different category, it should be going down. But no one, I've never seen anybody that's done although I've read thousands and thousands of papers and books on hybridization, I've never seen anybody that did anything like that. It's just that basic kind of experiment. Instead, they just say, oh, if it's more distant, it's just not going to work. And they just have heard that from someone else.
Yeah.
They
Yeah.
don't know it. They have no experimental data to back it up. And yet, it's just an example of scientists being unscientific. They just, uh, you can be a scientist and uh, gather all sorts of data and make all sorts of uh, careful and accurate measurements, but you can also just be repeating hearsay in certain aspects of what you're talking about. And that's just hearsay. There's no empirical basis for it. And
It's interesting too because
and so
you could imagine that as you move from like species, genus, family, order, etc., just out of the air numbers that it goes from 100% to 10% to 1% to
you can
0.1% success rate, but the number of species within a genus and a family and an order and a kingdom grows by like an order of
yeah,
magnitude
that's
or
an
more.
additional issue.
Yeah,
We
so
were talking about that. We were talking about that also before we started recording, and as I was mentioning to you, the number of combinations goes up as a triangular number, which is, say, if the number of organisms being paired. If you have, say, 10 organisms, then you have 10 times 10 plus 1 divided by 2, which is 55 different combinations from those 10 organisms. And then if you have 100 organisms, it's 100 times 100 plus 1 over 2, which is 5,000, which gives you 2,525, I think.
mm.
And so it's much, even just going from 10 to 100, you have a tremendously larger number of combinations. And then if you can imagine if you go up to a million, it's going to be a million times a million divided by two. So it's going to be like, what, half a trillion crosses that you can get, different
Now
crosses. And
there's
so,
a
and so that, like I said, that's a separate issue from what we were talking about before. But then if you impose that on that chart, if there is any sort of decline from right to left with an equal number of trials, uh, that would be probably, to my mind, more than compensated by the fact you have a much larger number of different crosses that you could potentially have out in the right-hand part of the chart.
And
So,
this is something that people who at least are interested in doing wide crosses in plants, one technique you can use to deal with the logistics, like nobody's got time to do 1,000 controlled labeled hand crosses. But what you can
mm,
do is you mix all of your wide crossing pollen together and apply it in combination onto the stigma of a limited number of plants. It increases your chance that some of those will be viable. And in the next generation, if you can identify what the successful pollen parent was from the hybrid,
yeah,
you then
that's a good
exclude.
way of like just taking a shot in the dark, I guess you'd say.
Exactly,
It's like,
exactly. And once you know, oh, this hybrid works and I have that material crossed, you take that pollen parent out of the mix next time and give other varieties a chance. Because usually if there's a variety that's more fertile than others, it'll kind of muscle everything else out of the way. It'll take that opportunity. And if you've got a much more remote chance of something else happening, you don't see it. So
yeah,
that's
well,
one
you're talking
strategy.
about the aspect of trying to actually produce something, where I was talking about from the aspect of trying to get a handle on that statement
Mm.
that people make that, if things are more distantly related, then you have a lower chance of the hybridization being successful. Um, and so that experiment that I was talking about was intended to evaluate that statement. And
Yeah, yeah, yeah. I was just taking it to people who are
but
thinking of trying the limits of wide crosses in plants. It's one way that you can juggle all of these options, all these possible combinations more efficiently if you give up on having to know exactly what every cross was. Because if 99% of them aren't going to work, you may as well do all of them at once on every flower.
yeah, and also like one thing is just to like keep your eye out. I found like, well, it's not extremely distant hybrid just in my yard today. I think that it looks to me like a, say, zinnia chrysanthemum cross,
Hmm.
which is rather odd. I've never heard of that. I suppose there may be such a thing out there, but it's this
There's been a
flower.
recent paper from some workers in Ukraine working with pollen-free strains of sunflowers, and they
Mhm.
found pretty much everything in the Asteraceae that they put the pollen onto those sunflowers. They got all these weird hybrids out. So yeah,
Yes.
Asteraceae is more broadly interfertile than people realize.
Yeah. Well, like I hadn't realized myself. Like I'm more of an animal person than a
Mm,
plant person, but the it didn't occur to me that a zinnia might cross with a chrysanthemum, but
mm,
that's what it looks to be. It's got a flower that's kind of shaped like a chrysanthemum, but it has the coloration is like the zinnias that are growing nearby.
mhm. Oh,
So
now
I had
you might be able to point me to a study that I remember reading long ago, and I've searched for it and I can't find it. It was quite old. I think it's from the early 20th century, and a researcher took the sperm and eggs of all sorts of marine king families, like really distantly related animals, and he mixed up the sperm and eggs of like starfishes with tube worms and corals, like things that should be very differently structured. And he was getting embryos developing from those really
No, I had that.
unimaginable.
I can send you that in an email. I don't have it at my on the tip of my tongue, but
But you have heard
I
of
know,
it.
I know what you're talking about. I know
Yep.
where to find it, so I can get that and send that to you. But
Yeah.
yeah, it was one thing I remember. He was producing starfish and sea urchin crosses.
Hmm.
Crosses between starfish and sea urchins, which is fairly distant. I think those are considered, might be a mistake, but I think those are separate classes that are
Hmm.
kind of. But so have we exhausted this particular aspect of this, or we need to, you want to ask more
There's
about
one
this?
final really provocative thing that I want to throw in. So, and it kind of relates to a thread that I was pulling at before, that living fossil species that don't seem to change for millions and millions of years often seem to be monophyletic. Like you've just got one type left. And if
Yeah.
hybridization is powerful for stirring up the genome and causing changes and that stepwise jump in evolution, that might be a reason why these isolated species just kind of paint themselves into a corner. They've got a niche where they can function, but they lose the ability to hybridize easily with something relatively closely related and like leapfrog into other niches. And it made me wonder if that has happened to humans, that modern humans have expanded so rapidly. And despite our superficial differences, and the absorbing of older populations, we're pretty much one type now. And it made me wonder if we might be forced to use wide hybridization to kickstart human evolution into some next distinct form. Is that a bit too Island of Dr. Moreau to be
Well, I
taken
think it
seriously?
might not go over well with some of the more conservative people here in the United States that are seem to be taking control of our lives now. But I think that, well, I think that there's a lot of that stuff that's going on already from what I see, the reports that I see just coming in still. I've seen hundreds of reports going back centuries and up right up to the present that, allege human animal hybridization. And but some of the more recent things are like I was saying, like there's did I mention it to you already? There's this guy in El Salvador that was reported earlier this year who was going over to visit the donkey next door that belonged to his neighbor, and the donkey gave birth to a human-like offspring. It's photographed in the news and stuff. And then the thing that I found surprising, uh, most surprising about that particular case was that he was fighting for his parental rights. He said that this was his child and that he was rightful owner and rightful parent, and that he should have the care of it, not this guy just because he owned the donkey. And so
It
there's
makes you wonder how if these hybrids are being created, how often the human that's involved kills them out of shame before
I
anyone
think that happens a lot, or
finds out about them.
shame might not be the right word. It might be fear,
Hmm.
fear that they're going to be punished.
Hmm.
Um, there was another recent, well, not that recent. I think it was just like 6 years ago now, but a woman from Hungary contacted me about a pig human that had a sow had given birth to there, and she sent me pictures of it, and she was trying to, this animal into her possession and was going to send me samples and so forth. And but the owner was freaked out by it all and didn't want to cooperate in any way and ended up killing the animal.
Hmm.
Or I'm not sure in that case whether animal is the right word. Um, but yeah, that I think that happens a lot. There's an extreme taboo associated with well, it's it's not only bestiality, which is just the sexual aspect of it, but this is bestiality coming to fruition, which is even more shocking and disturbing to a lot of people, I think.
It challenges human identity at its deepest level.
Yeah, it's like and so what I was saying earlier is that you were saying that it might be something like that necessary, something you were saying that something like that might be necessary to get humans out of their genetic doldrums or whatever they're like
Hmm.
the gotten isolated and stabilized so that they're no longer progressing or whatever. So that might be an option, but I do think just from these things that are reported to me, like Costa Rica and not Costa Rica, El Salvador and Hungary and all these other places, that stuff is even now still going on. And also that my book Telenothians, I have reports that I quote in there that go all the way back, to Roman times or earlier.
Mm.
I mean, the earliest cases in there are just representations, pictorial representations of various types of organisms. But I mean, like an early one was Alexander the Great's horse was supposedly a cow horse, and so that was something called a Jumart. And supposedly his horse had horns.
Hmm.
Uh, Bucephalus was his name, which means horse head. I mean, it means cow head.
Hmm.
Uh,
Do you
and
think we're just in a holding pattern until a definitive sample ends up being analyzed where it's incontrovertible? Like that, if and when that happens, the cultural scientific impact is going to be extraordinary.
Well, I think already there's a continuum of mindsets, and the same evidence to different mindsets implies different things. For me, what I've seen over my lifetime, the stuff I've written about in my various publications, all this information I've collected, it's indicates beyond doubt to me that these sorts of things do occur.
Mm.
But to someone else who just hears a couple of cases and doesn't really want to believe it anyway, they might not believe it.
Mm.
And then other people that will never hear about it at all, and they'll continue believing as they have been. They're not curious about it. They don't want to inform themselves about it, and they will continue merrily on their way.
You're right. Like
Um,
even solid evidence can only do so much to change people's attitudes because you can always doubt the quality and the veracity of the report that reaches you,
or the
on the other.
reporter. I
Yeah,
mean, a
yes.
lot of people, like essentially attacked my character, saying that I was like some sort of nut for saying that such things could even be possible, where for the most part, I'm just quoting existing reports, but the fact that I'm quoting the reports to them strikes them as nutty. But I mean, what else can you do? You can't find out about something if you can't even read the report.
Yeah, yeah.
You cannot find out about it. And so,
Oh, I had one. I
so,
had
so
one
for those people, so
question.
those people, the knowledge is sealed off. They can't reach it.
Yeah, yeah. I did have one question. Your recent paper on the genomic analysis of pigs and bonobos and humans that found
Hmm.
that there was a pattern in the 1.5%, I think, of human DNA that's different from bonobo DNA.
I believe the number mentioned in the paper, I believe, is 1.3.
1.3,
But
yeah. But yeah, that there's correlations between that changed DNA and pig genomes. There was a question I had from a comment someone I saw saying if the analysis could be repeated with non-pig animals to see if this is like just an artifact of the method of analysis or if there's a genuine signal. Are there any
well,
possibilities or plans to do that?
well, as I was mentioning to you before we started recording, that paper is meant to address objections I've encountered in connection with other lines of research relating to that topic. In other words, I found long ago, I mean, many years ago, that the human features that distinguish us from bonobos, chimpanzees, and for the most part from all other primates are very consistently found in pigs.
Hmm.
And so my hypothesis for explaining it was there was some kind of pig ape hybrid. But the objection that I was had mentioned is that people said, well, if that's true, then there should be some sort of genetic trace of that. And so I looked and I looked and I couldn't find, even though I'm a geneticist, I couldn't find what that trace might be. I mean, I looked at all sorts of different genes. For years I was looking for different kinds of genes, but the problem was I didn't know what I was looking for. I didn't know what genes to compare.
Hmm.
You can't just like say, oh, well, these are going to be the pig trace genes. You have to just look at one gene after another, after another, after another, and kind of a blind search and hope that you could find something. But the thing that the paper reports is I took more than 800,000 different randomly selected pig 40mers, nucleotide 40mers, which means like nucleotide sequences that are 40 nucleotides long.
Yeah, 40 letters
And
long. Yeah.
put them in a big file, and then searched with that large set of randomly selected pig 40mers, searched both the human and the bonobo genomes, which they're in the theory that I proposed. The bonobo or something like it was one of the parents that mated with pigs to produce proto hominids that back crossed with the bonobo side a couple of times so that we ended up being more like bonobos.
Hmm.
And that out of that population, the human race stabilized as what we see it now. And then perhaps it's really still stabilizing, I would think. But the thing is that under those circumstances, you expect to have, if you're taking these 800,000 40mers, get a little bit higher rate of match with humans, since they're pig,
Mm.
than with bonobo, since the bonobo has no expected trace under that hypothesis. And what I found, I was just hoping to see, that there might be more in humans. But what was remarkable to me is the amount that I found more indicated that virtually all of the nucleotides that distinguish humans from bonobos match pig.
Hmm.
Those nucleotides match pig. The 1.3% is all it is, but those 1.3%, which amounts to many millions, I think it's on the order of 70 million nucleotides,
Mm.
uh, match pig in the human genome. But so, in other words, there is this extra 70 million or so nucleotides in the human autosomal genome that match pig. And so that's consistent with that hypothesis. It doesn't prove that hypothesis. Could be,
Yeah. Yeah.
you could also you could run the same thing if you took say a baboon and you used it instead of pig. You would get much higher hit rates with baboon than you would with pig
Mm.
because humans, under the hypothesis, at least this is the way the hypothesis would explain it, is that the humans are back crossed to primates, and so they're more primate-like. And
Mm.
so, since baboon is a primate, it's going to uh, jack the rate up at which it gets hits. So,
There's
um,
also the mystery that humans have two fused chromosomes compared to bonobos. And if you
they have
go for
what?
the
Two what? They have two what?
two,
Fused? Did you
two,
say
yeah, two two of our,
F-U-Z-E?
two of the bonobo chromosomes fused in humans, so we've got a slightly different chromosome number. And if you believe the hybrid incompatibility story, like with mules, because horses and donkeys have slightly different chromosome numbers, so therefore mules are sterile and therefore hybrids are unimportant. That raises the question of how did humans derive from a bonobo-like ancestor? Because the first human with a different chromosome number would have been infertile, crossing with any of its other compatriots with the larger chromosome number.
Well, is this something you're asking for someone that listened to one of the other podcasts?
Well, no, it's more just it's a very simple thing that you can highlight that the usual
Well,
story that, with horses and donkeys and mules and chromosome numbers is too simple. It doesn't work when you think it through the
well,
consequences of human evolution from a
well,
bonobo-like ancestor.
just a general way I can say, Shane, is that the hybridization and produces organisms that are not, as a rule, as fertile, as they don't produce as many gametes
Hmm.
as the two types of organisms that cross to produce them. But they do produce gametes. They produce lots of gametes, and they produce, in many cases, a lot of deformed gametes. But there are many cases of hybrids producing offspring. In my book that I did for Oxford University Press on hybridization of birds, it's like a worldwide survey of, essentially, I try to get all reported types of avian crosses and all of the papers in which those had ever been reported into that book. And it amounts to more than 4,000 different kinds of crosses. And just as a matter of curiosity, I went through to see what, this is not a formal study, this is just something I did on my own.
Mm.
And I never really reported, but I looked to see how many of those crosses first um, there was any indication that that type of hybrid had been able to produce offspring. And then also whether there's indication whether they were highly sterile and were unable. So those two categories, either that essentially information about whether that type of hybridization was sterile or not,
Mm.
absolutely sterile or not. And so that can go into the heading of if they produce offspring, definitely they're not absolutely sterile. Or it could come under the heading of there are many papers out there or reports saying that they could never produce offspring with this type of hybrid. And so the ones that were in the former category, the crosses that had information indicating that they had produced offspring, outnumbered the ones that were definitely established as being highly sterile by a ratio of 8 to 1. It's like eight
Mm.
times as many crosses they were able to produce offspring, the hybrids. Not just the parents producing hybrids, but the hybrids themselves could produce offspring
Yeah. Yep.
8 times more commonly than they were reported to be highly sterile.
What,
So
where do you
it
see the
depends.
potential in biotechnology of hybridization versus more narrow manipulative techniques like CRISPR, genetic engineering? Like, do you have any broad thoughts about where those are going to take humanity in the future?
Well, I think hybridization has already been used for like millennia, like, to produce things that are valuable for human beings. And some of it's serendipitous, and or some of it has been directed, like what you were talking about, or you're doing
Hmm.
these mass pollinations to see what you can get out of it. Um, but the CRISPR, that's not something that everybody can do.
Hmm.
I, almost anybody could, if they want to spend the time and set up the situation, they can do hybridization. But you have to be highly educated in a particular field to be able to do CRISPR. You have to have a lot of technical expertise. And so, and then there's the question of, you're still going to have to experiment around in that case to see what it is that you can actually produce. So I don't know. I think that I wouldn't want to, if you ask me to say which would be better, I wouldn't want to discount CRISPR completely, but obviously I'm heavily biased towards hybridization. I might not be the right person to ask that question because for me, it's like, no, who would want to do CRISPR when they could do hybridization?
Yeah. The big weakness I see with CRISPR is that it can only do what you know you want it to do at best. So, for example, if you know this particular gene makes this particular protein, which has this particular property, fine, you can do that with CRISPR. Exactly where you drop that in the new genome is still a bit of a crap shot. Like we don't fully understand why a gene in one location is expressed well and in other places it's not. There's all sorts of levels of control and architecture going on there that are a bit of a black box.
Mhm.
But that means that you're limited to genes and proteins that you can understand and that have simple functions. I mean, there's for example, for like secondary metabolite production, for making like an alkaloid, there can be a set of genes that all work together in a fairly well characterized pathway, but there's so many cross-talking networks and possibilities in biology that I don't know, CRISPR feels like it's a tool that's really limited by our lack of understanding about the totality of biological systems. Whereas something like hybridization, it's much more open to serendipity. Like you're moving, tens of thousands of genes, rearranging those networks when you do hybridization. And it seems like biological systems are designed to be able to cope with those disruptions quite well. Like it's amazing that you can throw those two sets of organisms, chromosomes together, and how often it still functions. It still kind of limps along. So yeah,
Yeah, and also there's it kind of, I don't know, I guess I kind of think of it as self-screening because, you say you take like a million spermatozoa and put them on 1,000 eggs, it may be that 99% of them come to nothing, but you still end up with 10 organisms, say, coming out of that
yeah,
mess.
And you don't
So,
need to understand in advance why that 1% of combinations works. You just need to
yeah,
do enough experiments
yeah,
to make it work.
right. But the CRISPR or any other biological um, type of biotechnology, I don't want to discount it.
Mm.
Uh, it's just that I think that it it requires a level of education and technical expertise that's going to limit the number of people that can be engaging in it.
Yeah, yeah, yeah.
Um, like I, some of these guys that I went to graduate school, they're just, geniuses that really just know all the details of how the molecular interactions are occurring, and they can come up with clever new things to do with that. But that's not something that everybody can do.
Hmm.
I think that's the big advantage that hybridization has, is that virtually anybody can do it. All you have to do is put a couple of animals into a pen together and then see what happens. So it's nothing more technological than that in a lot of cases. It's just, I mean, a lot of it just happens just by accident, even. Like there are many cases where, say, it's zoos, they've set up a bunch of birds in the same aviary and they say, oh, wow, look at this. We didn't expect to get that. There's a
Hmm.
strange combination here. And then nobody's ever heard of it before, but they got it. And then in a lot of cases, these birds and animals, plants, I think you were saying, like they can have advantageous characteristics that, will be useful to agriculture or, various other human applications. So,
We should probably start wrapping this up because we had some very long chats before. I want to keep this one to the usual kind of hourish long. But I did want to ask you one kind of open-ended question. Do you think there is a group of organisms that has potential to be transformed through hybridization that humanity has like barely started even considering as something that we could put to use?
uh, uh, I guess I haven't really thought about it in those terms, so I don't know what to say.
You can take your time, like think it through.
uh,
Let's focus on the animals because like this show normally gets lots of focus on the plants, and you're more of an animal expert. So I like
Well, just in general, I don't know if I would say that there's a particular category that has more potential for having interesting things come out, but just with animals, as I was mentioning earlier, there's certain types of animals that have much more of a propensity to uh, mate with other kinds of animals. Like I mentioned before, chickens, pigs, dogs, ducks, and like also say canaries. Canaries will have been reported to mate with many different kinds of birds. And so if you were to work with a particular one of those, say five that I just mentioned, you'd be much more likely to easily obtain something new just by pinning those animals with some other kind of animal. It would be like a small amount of labor
hmm.
involved. Just put a female and a male together and then wait and see what happens. So it would be, I mean, smaller animals would be more economical, but even the same sort of approach would work with larger animals. Also, cattle have been crossed with many different kinds of animals,
Mm.
but it depends more on like how much money you have to set up your, experiment setting. Um, but you could try many different types of combinations by those means. And I imagine also with plants, there are an analogous situation, ones in which you can easily get at the pollen, easily apply it to the flower to carry out your fertilization, or there might be systems where you can just like blow the pollen across en masse onto recipient flowers.
Hmm.
Um, I mean, it would be the cases I guess you'd say, where little effort was required to make it occur,
Yeah.
where you could get the most out. Say that's one reason why there's been more experimentation in hybridization with fish than there has been with mammals. It's because you can just squeeze the milt out of many different kinds of fish and then put it on the eggs, where you squeeze an animal, a mammal, hard enough to get the sperm out. It's not going to be a very happy mammal.
Doesn't exactly put them in the mood for love.
Or maybe it doesn't even put them in the mood to survive. But yeah, I would just say, generally speaking, the thing to look for if you really want to do a lot of experiments at random is the easy systems, the ones where the fertilization can be easily accomplished,
Hmm.
artificially, or even just by putting the animals in together. And also, with animals, say it's important to deal with ones where they can be easily imprinted. That is, imprinting is the phenomenon where if you have an organism raised by some other kind of organism, it will want to mate with that type of organism when it reaches maturity.
Hmm.
Like say, if a goose is raised, there's like, I think it's during the first 48 hours of its life after hatching, there's around, say, a turtle. It will follow the turtle around for the rest of its life and try to mate with it. And that's a sort of situation where you can set up your experiments very easily and end up having to do very little work yourself.
Yeah, yeah. Well, that's what this show is all about. It's to open people's eyes to the possibilities that are sitting right in front of them, that are accessible for anybody with a garden or a little bit of space, maybe even like a tissue culture lab if you set it up in an apartment. There's all this possibility for people to play around with. You don't have to wait for people with $1 million machines in laboratories to do it.
Mhm. Yeah, so I'm interested to hear some of the results of some of these things. I'm always curious about all this sort of this whole phenomenon of hybridization. But we'll just have to see. Like, there's no telling, like all sorts of things that we've never heard of that I think could pop right out of it.
Hmm. And just out of interest, I'll plug my damn book again, Taming the Apocalypse. If people want to check it out, it has a survey of the entire tree of life for organisms with domestication potential, and usually looking at routes where hybridization would be possible to kick off a little mini revolution.
Yeah, well, I mean, in that case, I can see in a lot of cases just by crossing with some sort of domestic animal that had desirable domestic traits, you could bring these other organisms into the domestic sphere more easily with their hybrid offspring.
Mm,
If you see what I'm saying? It's like,
yeah.
say with animal, like if you didn't want it to be wild, you wanted it to, be more docile or whatever the domesticated trait is that you wanted to have, you could bring those traits into it by means of hybridization. There's a widely used method, introduction of traits that uh, lots of breeders have used. They just want to bring particular traits over from one type of organism to another, and they just uh, cross it with some organism that has those traits, and then they select for the offspring that have that trait, and it's introduced into the new setting. So,
With animals too, it's remarkable how quickly a domestication syndrome can be selected. The Belyaev foxes in Russia that went from being like wild animals that wanted to bite your face off to puppy dogs that like wanted to climb all over you and lick you. That was done in 5 generations of very non-technical selection. And that process has been repeated with rats and mice in laboratories. I'm trying to think of other examples of that. So yeah, even though these things like we think of the domestication of dogs or cattle, even though it happened thousands of years ago, that doesn't mean it took thousands of years to happen. It probably happened within a small number of generations with the right pressures applied.
yeah, well, we know, like in the case of like bread wheat, that was just like very compressed. It was just like to produce a polyploid like that, it's just a couple of generations.
Hmm.
So, I mean, I, I believe there was a two-stage thing where they produced an initial hybrid at some stage and they were growing that, and then they had a second hybridization event at some point that turned
Hmm.
it into a triploid, or not triploid, but it was a hexaploid.
Yeah, yeah.
And, And and so those, when you actually look at what the process is with that sort of thing, it's usually just something that takes a couple of generations. Excuse me.
Mm.
And so that's the thing that I think one of the major features of hybridization is the rapidity with which it can occur,
Yeah.
the,
Yeah.
the huge alterations that occur in very brief order.
And it's
Brief,
definitely
brief.
something that a single person can do in a single lifetime, as long as you're not working with like blue whales or sequoia trees.
That's something I should have mentioned earlier. Like generation time is important too if you want to. Yeah, I don't think you want to breed new types of whales or sequoias. That could be a little bit.
You'd be waiting a while. Well, on that note, thank you very much for coming back to talk again to tease out where this hybridization theory could take us in the future. It's been fascinating, and I'm so grateful for you taking all the time to talk again. So, before we go, can you once again let people know where they can learn more about your work and various books of yours that they can check out?
Well, the main place to find information about what I've been doing is on macroevolution.net. There's a lot of different stuff there, but I have a on origins of new forms of life, which is the theoretical presentation of stabilization theory and how hybridization fits into the evolutionary process. That's what it talks about, and that's on there. That's a back in 2007, I submitted that book to Oxford University Press, and it was accepted. I was under contract for about a year, and then because it was I uh, contradicted Darwin in a lot of respects, uh, I got flack for that, and they were insisting that because it contradicted Darwin that I change it, but I didn't change it. Instead, I just said forget it. I'm just going to make it available, and so it's available there. Um, but there's that. And then there's also lots of information on there about very distant reports. There's mostly just quoted reports of very distant kinds of hybrids, interordinal hybrids, or even interclass hybrids that have been reported in various places over the last several centuries, right up to the present. Lots of videos, pictures, news reports, scientific reports. A lot of them are translated from other languages. I've translated hundreds of reports from Latin, German, French, and also from Spanish and made them available there. And I've sold all the copies of this book, Telenothians, that I compiled, where I compiled like distant crosses. All the crosses in it are like interordinal or more distant,
Mm.
all the reports.
I've bought a copy and checked it out, and it's eye-opening. It's so extraordinary how much work you've done gathering all of that information together.
And now I've got like a third edition where I've doubled the number of reports that were in that.
Wow.
Uh, so there were 1,500 approximately in that, and now there are 3,000 and it's new. And the new edition, which I'm hoping to get out soon, although I'm kind of depressed about all the political events here in the United States, but I'm
Hmm.
trying to uh, muster up the momentum to take that to the finish line. And I, one thing I have in that wasn't in the previous editions is I've got a complete chronology from like, about 1000 BC to the present of every reported interordinal or telenothian event
Hmm.
in the location at the back, so that it's all just compiled so you can get an idea of how many different kinds of each different kind of telenothian there might be. The most common one in there, 300 out of 3,000 reports, is pig human.
Hmm.
That's the most common one. And then there are other ones that are nearly as common, but then there are other ones that are, sorry, I'm getting a little bit hoarse
That's right.
all this talking, but there's some of them there that are just one report ever. It reminds me of my bird book where I had certain types of bird hybrids. There had been one specimen reported ever,
Hmm.
you know, like such and such a hybrid back in 1894. It's the last one was ever seen. And so it's the same thing with the Telenothians. There's certain ones that are very rare. But I've got a more extensive synthetic section at the back where I enumerate all the different kinds of hybrids, make pie charts and so forth, so that you
Hmm.
can see all the different kinds, and also the ones that are more fertile, and talk about all the different aspects of the various interesting topics that I have pursued. Like one I find kind of interesting are the various human births where they look like they have big patches of animal fur here and there, or sometimes over large parts of their body. It's called hypertrichosis. Also, they call them hairy. So there's a whole chapter on reports like that and lots of pictures. So it's to me, it's definitely after compiling and writing all this up, it's a reality. But I
Mm.
can understand why if someone's just hearing it, say on the podcast here, or someone that's unfamiliar with reports that I've read would think, well, that's just silly and impossible. I can understand why they would think that. I thought the same thing myself about a lot of these things maybe 10 years ago. So what makes a difference is encountering the reports, reading them, and seeing the evidence. Once you read reports and see the evidence, then your outlook shifts.
Mm-hmm.
Uh,
Mm-hmm.
that's just the
And
way
yeah,
it goes. I think that's one thing. I
I
think
hope
it's the
this I
thing.
hope this show I hope having you on the show has helped push that idea out into the world a bit more in a friendlier light, because I definitely think it's a concept whose time has come and it needs more serious investigation and consideration.
Yeah, well, that's been my hope in all of this is that it just be a springboard for further research. That it's like something that like when I first started looking at it years ago, I don't think that I think almost no one was looking at it at that time. I just got curious about it, and it was just Darwin was the one that got me going on. He's saying that there are all these hybrids that were able to produce offspring, and in the Origin of Species has a whole chapter on that. And I
Hmm.
was surprised that hybrids could produce offspring, and so it got me curious. And I started reading more about hybrids, and so I've been curious about it ever since.
Oh, hopefully on that note, we can leave people curious to explore this avenue for themselves in the future. So,
Well,
Eugene,
obviously, I highly recommend it.
yeah. Okay. Well, thank you for the incredible amount of time and thought and consideration that you shared with us. And yeah, I'll be very interested to see what kind of feedback we get from the audience when these episodes go out.
Well, let's do it again soon before I have to flee the country here. All right, bye bye.
Bye.